Genuinely excited for this one.
Me too. This is where it's really strange.
It really does. We spend so much of our lives just observing. We're looking at screens, at faces, navigating the world, and it all feels so seamless, so automatic.
Right.
You open your eyes and boom, the world is just there.
It feels effortless, doesn't it?
Yeah.
And that's maybe the greatest trick the brain ever pulled.
Exactly. It feels like this passive activity. But looking at the research we're covering today, specifically Chapter 52 on the central neurophysiology of vision, it turns out that seeing is probably one of the most complex, active, and, energy-intensive things your body does.
Oh, by far. And we're not just talking about the eyeball today. We've covered the optics, the lens, the retina.
We did all the hardware.
Exactly. Today is about the black box. It's about what happens after the photon hits the retina. And in a way, the lights go out.
Right. We are decoding the central neurophysiology of vision. And looking at the stack of notes here, I think the best place to start is with a bit of a mind bender, just to set the stage.
I think I know where you're going with this.
I want to start with this premise. Seeing doesn't actually happen in your eyes.
That is technically, and I think profoundly correct.
Your eyes are just the lens. They are the hardware gathering the data. They're the webcam that's plugged into the computer.
Mm-hmm.
But the brain is the photographer, it's the editor, the color grader, and it's the screen all at once. The image you think you see, the face of your friend, the words on this page is actually a reconstruction. It's happening in total darkness inside your skull.
That is the perfect way to frame this. Think about it. The retina captures photons. That's just physics.
Right.
But those photons are immediately converted into electrical impulses. From that point on, everything we call vision is just electricity zipping through wet
Or
wires.
sparks.
Just sparks. And it's up to the central nervous system to turn those sparks back into a 3D, colored, moving reality. And the source material today, essentially gives us the wiring diagram for how that miracle happens.
So our mission for this deep dive is to trace that physical journey. We are going to follow the path of a signal from the moment it leaves the eye until it, explodes into your conscious mind as an image.
It's quite a path.
It is. And looking at the roadmap, this is a dense topic. We've got neural highways, we have gatekeeper nuclei, cortical layers, and some pretty serious autonomic engineering to get through.
Yeah, so maybe we can break it down sort of into 4 main acts just to keep it organized.
Okay, let's do it.
First, we can talk about the highway, just getting the signal from the eye to the brain via the visual pathways.
Okay.
Second, the gatekeeper, this fascinating structure called the lateral geniculate nucleus, or LGN.
The LGN, got it.
Third is the processor, the visual cortex itself, where the real analysis begins. And finally, we can cover the mechanics, how the brain physically moves the eyes to control what data it's even getting in the first place.
That sounds like a solid plan. Let's hit the highway, segment 1, from retina to brain.
All right. So imagine you are a signal. You've just been generated by a rod or cone in the retina. You start traveling down the optic nerve.
Okay.
Now, you look at the diagrams in our source material, especially figure 52:1.
Mm-hmm.
You see the first major junction, the optic chiasm.
The chiasm, this is that famous X-shaped crossing point at the base of the brain. You see it in every anatomy textbook. It just looks like 2 cables merging and then splitting again. But the wiring here isn't just a simple crossover, is it? It's very specific.
Oh, it's incredibly specific. And it is absolutely vital for how we perceive the world as a single thing.
Right, because you have 2 eyes, but you don't see 2 separate worlds. You see one unified field.
Exactly. The optic chiasm is where the sorting happens to make that possible.
Okay, let's unpack that sorting mechanism, because the text makes a big distinction between the nasal halves of the retina and the temporal halves. I think we need to visualize this carefully for everyone so they get why this crossing even happens.
Okay, good idea. So just visualize your left eye.
Yeah.
The retina is that curved bowl at the back of the eye, right?
Right.
The nasal half is the side of that bowl closest to your nose.
Okay, inside.
And the temporal half is the side closest to your temple or your ear.
Got it. Inner half is nasal, outer half is temporal.
Now, here's the physics part. Because of how the lens in your eye works, remember, a convex lens always reverses an image.
Flips it upside down and backwards?
Exactly.
Yeah.
So light from the left side of your visual field actually hits the nasal part of your left eye.
Okay, hold on. That makes sense. The lens flips it. So light coming from my left crosses through the pupil and hits the right side, which is the nasal side of my left retina.
You got it. And by the same token, light from the right side of the world hits the temporal part of the left eye.
So the image on the retina is already flipped left to right before any nerves get involved.
Precisely. Now here is the magic of the chiasm. The fibers from the nasal halves of the retina, so the ones seeing the outer edges of your vision, they cross over to the opposite side of the brain. They decussate.
Decussate, that's a great word. It just means to cross in the form of an X.
It does. But, and this is the key, the fibers from the temporal halves, the ones seeing more of the center field, they stay on the same side. They do not cross.
Okay, so let's trace this through to the result. Why does the brain do this complicated partial split? I mean, why not just send the whole left eye to the right brain and be done with it?
Because of the overlap. The result of this very specific sorting is that all the signals from the left visual field, it doesn't matter if they came from the left eye or the right eye, they all end up in the right side of the brain.
Wow.
And everything from the right visual field ends up in the left side of the brain.
That is wild when you really stop and think about it. The brain literally just splits the world down the middle.
It does. It cuts your reality in half vertically and then processes each half with the opposite hemisphere. This is why, for example, if you damaged your left optic tract after the chiasm,
After the split.
right, you wouldn't go blind in your right eye. You would go blind to everything on the right side of the world in both eyes.
That's a distinction that trips a lot of people up. Blindness in an eye versus blindness in a visual field.
It's a critical difference. And once they pass the chiasm, these bundled fibers are now called the optic tracts.
They're not nerves anymore.
Right, because they're now inside the central nervous system. And these tracts carry that sorted information to a very specific spot in the thalamus.
Which we'll get to in a second. But before we even enter the thalamus, the source material points out something I hadn't really appreciated. I always just assumed the optic nerve plugs straight into the visual cortex, but not all the nerve fibers go there. Some of them split off to what the text calls the old system.
Yes, this is a fascinating evolutionary relic. We, human beings, we have this massive, sophisticated new system for conscious vision, for reading, recognizing faces, seeing color.
Right.
That system goes to the cortex via the thalamus. But underneath that, we still have this old system that directs signals to the midbrain and the base of the forebrain.
So this is like our lizard brain vision.
Essentially, yes. It's the visual system that, say, a fish or an amphibian relies on almost exclusively. They don't have a massive visual cortex like we do. They rely on these older centers.
And the text highlights three main targets for this old system in us.
It does.
Right.
First, the suprachiasmatic nucleus in the hypothalamus.
That's a mouthful. Suprachiasmatic. So above the chiasm, what's its job?
It's your master body clock.
Yeah.
It uses visual data just to know if it's day or night. It controls our circadian rhythms.
So it doesn't care what you're looking at.
Not at all. It doesn't care if you're looking at a painting or a brick wall. It just cares about the photon count. Is it bright outside? Okay, suppress melatonin. Is it dark? Okay, release melatonin.
So even if you were consciously blind, like if your visual cortex was destroyed, this part of your brain might still know when to wake up.
Exactly right. As long as the eye and the optic nerve are intact, your body rhythm stays synced to the sun, even if you see nothing but blackness.
That is incredible resilience.
But-
Okay, what's target number 2?
The protectal nuclei in the midbrain. These guys are responsible for reflexes. Specifically, focusing the eyes and that pupillary light reflex making your pupils shrink in bright light.
Ah,
Again, this happens without you consciously seeing anything. It's a pure hardware reflex.
like a camera's auto exposure. It doesn't need to know it's taking a picture of a sunset to know it needs to close the aperture.
Perfect analogy.
Hmm.
And the third one is, I think, the coolest. The superior colliculus.
The superior colliculus, it sounds like a Roman general or something.
It kind of acts like one.
Mhm.
In primitive animals, this is their main visual center. For us, it's responsible for rapid directional movement. Let's say you are walking down a dark street. You see a sudden flash or a movement way out in your peripheral vision. Your head snaps toward it before you even know what it is.
Right. I've definitely done that. I've turned my head before I even realized I saw something.
That's the superior colliculus. It detects a disturbance
Hmm.
and it directs the eyes toward it so the new system can get a better look and analyze it. It's like a security guard scanning the monitors who alerts the detective when something moves.
So it's a security system just running in the background.
Exactly. It's always monitoring for threats or sudden changes. But for the rich, detailed, technicolor movie that we experience as reality, we need the new system.
Mm-hmm.
And that brings us back to that junction in the thalamus, the dorsal lateral geniculate nucleus, or LGN.
Segment 2, the gatekeeper.
I love that name for it. The LGN is a small, curved body. Geniculate actually means knee-like. It's located at the dorsal end of the thalamus.
And its primary job is to be a relay station, right? It takes the info from the optic tract and then shoots it up to the visual cortex via something called the optic radiation.
That is function number one. And it's a high-fidelity relay. The source really emphasizes that point-to-point spatial transmission is preserved perfectly.
What does that mean exactly?
It means that if a photon hits a specific spot on your retina, that spatial location is maintained perfectly through the LGN all the way to the cortex. It doesn't get jumbled up.
So it keeps the map intact.
Yes. The top of the retina maps to the top of the LGN, which maps to a specific part of the cortex, and so on.
But, you know, if it were just a relay station, it would be a pretty boring piece of anatomy, just a wire connector.
Right.
But the text says it has a second major function, gating control. And this is where I had my first big aha moment of this research.
This is where it gets really, really interesting.
Mm-hmm.
The LGN doesn't just pass the signal along. It decides how much signal to pass. It's a filter.
It's a bouncer.
It is a bouncer. And here is the surprise. Where do the orders for the bouncer come from? The text lists two sources. One is the reticular areas of the mesencephalon, which is basically just an alertness signal. But the other is the visual cortex itself.
Wait, what? So the visual cortex, the place that receives the image, is sending fibers backwards down the line to the LGN to tell it what to send up.
Precisely. They are called corticofugal fibers. Fugal, meaning fleeing or moving away from the cortex. And these fibers are almost entirely inhibitory. They tell the LGN to turn down the noise.
So the brain is effectively saying, hey, I'm trying to focus on this one specific detail. Stop sending me all this other background data.
You've got it. Imagine you're looking for a friend's face in a huge crowd. Your cortex sends a signal back down to the LGN saying, suppress the signals from the periphery, suppress the color green, boost the signals that look like faces from the center. It highlights specific visual information by suppressing the rest.
That creates a loop. The brain sees something, decides it's important, tells the LGN to filter out everything else, which then makes the brain see that one thing even clearer.
Feedback loops are the secret sauce of neurophysiology. This allows the brain to direct its attention. Without this gating, you'd just be overwhelmed by visual stimulus constantly. It filters the feed before it even reaches your consciousness.
Wow. Okay, now we also need to talk about the internal structure of the LGN, because it's not just a blob of cells. It has layers, 6 of them, and they're very, very specific.
Right, the layers. Layers 1 through
Right.
6. And they're divided up by which eye they listen to and also by what kind of signal they handle.
Okay, let's break down the eye division first. That seems simpler.
It is. So layers 2, 3, and 5 take signals from the lateral or temporal, half of the retina on the same side.
Okay.
Layers 1, 4, and 6 take signals from the medial or nasal, half of the opposite eye.
So the zipper effect starts right here. Left eye or right eye, left eye.
Correct. But the more functional distinction, the one that really matters for how we experience vision, is the difference between the magnocellular and parvocellular layers.
Magno and Pardo. It sounds like a comedy duo from the 1920s.
They are the duo that makes vision work. Layers 1 and 2 are the magnocellular layers. Magno comes from magnum, meaning large. These contain large neurons.
And what's the specialty? Why do you need big neurons?
Speed. It's all about speed. Large neurons conduct electricity faster. These layers get their input from the type M retinal ganglion cells. These pathways are incredibly fast. Very rapid conduction.
That there's a trade-off, right?
Big trade-off. They're colorblind. They only see in black and white. And their spatial resolution, the detail, is very poor.
So the magno layers are low-res, black and white, but super, super fast. They tell you where something is and if it's moving.
Exactly. If a baseball's thrown at your head, it's the magnocellular layers that save your life. They don't care that the ball is red or has stitching on it. They just care that a large object is moving rapidly toward coordinates X, Y, Z.
Okay, so that's layers 1 and 2, the emergency system. What about the rest?
Layers 3 through 6 are the parvocellular layers. Parvo means small. Small neurons getting input from the type P ganglion cells.
And these are the opposite, I'm guessing.
The complete opposite. They transmit color. They've incredible point-to-point spatial accuracy, very high detail. But they're slower. This is your wet system. It tells you the texture, the color, the fine detail. It's what allows you to read the fine print on a contract or appreciate a painting.
So even before the signal hits the main brain, the image has literally been split into 2 separate streams. A fast black and white motion stream and a slower detailed color stream.
And that separation continues all the way up. Which brings us right to part 3, the processor, the primary visual cortex.
Also known as visual area 1 or the striate cortex, and it's located in the occipital lobe at the very back of your head. Specifically, the text says, in the Cowker fissure.
Mm-hmm. And the anatomy here is really important for understanding what happens when things go wrong. The signals from the macula, the very center of your vision where you have the highest focus, terminate right at the occipital pole, which is the very back tip of the brain.
And the periphery.
Peripheral vision is mapped further forward inside that fissure.
The text also mentions something called cortical magnification. This seemed like a really key concept for understanding how we prioritize visual information.
This is huge. I mean, literally huge. The fovea, that tiny little pit in the center of your retina that's responsible for all your sharp vision, it takes up a tiny fraction of the retinal surface area, less than 1%. It's pinhead size.
Right, it's minuscule.
But in the primary visual cortex, the area of the brain dedicated to processing signals from the fovea is massive. It has several hundred times more representation than the peripheral areas do.
It's like if you looked at a map of the world, but New York City was drawn the size of Texas, because that's where all the activity is.
That is a perfect analogy. The brain allocates its processing power based on importance, not physical size. We prioritize the center of our gaze above absolutely everything else.
That's why you can read a book, but you can't read a sign that's standing 3 feet to your left without turning your head.
Exactly. You just don't have the computing power allocated to that part of your visual field.
It's actually surprising how bad our peripheral vision is when you test it. We think it's good, but it's mostly just blurry shapes and motion detectors.
It really is. Your brain just kind of hallucinates the details based on memory to make you feel like you have this wide, clear field of view. But you don't.
Now let's get into the weeds of the cortex structure. Like the rest of the brain, it has 6 layers. Why is it always 6 layers?
It's the standard architecture of the neocortex.
Mm-hmm.
It's a very efficient design for handling input, processing, and then output. And those fibers coming from the LGN, those magno and parvo fibers we just talked about, they mostly terminate in layer 4.
So layer 4th is the inbox.
Is the inbox. And even here, that separation is maintained. The rapid magno fibers end in a sublayer called IDC alpha, and the detailed parvo fibers end in layer IDC beta.
So they still haven't mixed yet.
Not yet. From layer 4, the signals get passed up and down vertically. The visual cortex is organized into millions of these vertical columns. Each column is a functional unit, about 30 to 50 micrometers wide, with maybe 1,000 neurons inside. You can think of them as individual microprocessors.
And within this structure, the source mentions these things called color blobs, which is just a delightful scientific term. I thought it was a joke at first.
It is wonderfully descriptive, isn't it?
Yeah.
These are literal clusters of cells that are interspersed among the columns, and they are specifically activated by color signals. This is where the brain really starts to decode the wavelength information from that parvo stream.
So they actually look like blobs.
They do. If you stain the tissue with a specific enzyme, cytochrome oxidase, they actually show up as these dark blobs against a lighter background.
That's amazing. There's another piece of architecture in layer 4 that explains how we see depth. The text calls them ocular dominance stripes.
Right. Remember, up until layer 4, the signals from the left eye and the right eye have been kept completely separate. In layer 4, you suddenly have these interlaced stripes of neuronal columns. One stripe gets input from the left eye, the next stripe gets input from the right eye. They're alternating, about 0.5 millimeter wide.
So it's like a zipper, left, right, left, right.
Exactly like a zipper. And this is the very first time the brain can compare the two images side by side. This registering of the images is what allows for stereopsis, or depth perception. The brain looks at the slight difference between the left stripe and the right stripe, and uses that to calculate distance.
We'll dive deeper into depth perception later, but I want to move to segment 4, analyzing the image. Because the source material asks this fundamental question: what does the brain actually detect?
This is the part that really changes how you look at the world. You might think the brain detects light.
Yeah, that seems obvious.
But it doesn't. Not really. The primary visual cortex doesn't really care if a room is bright or dim. What it cares about is contrast, gradients, edges. The source creates this scenario. Imagine you're looking at a completely blank wall. It could be a brightly lit white wall or a dim gray wall. In both cases, only a few neurons fire. The brain is bored.
Okay.
Now, you place a solid black cross on that wall. The neurons that represent the middle of the cross don't fire much. The neurons for the background don't fire much. The neurons that go absolutely crazy are the ones representing the border of the cross.
So why is that? Why ignore the middle?
because all the information lives at the edges. If an area is uniform, there's nothing new to learn. The brain is an efficiency engine. It's optimized to find boundaries. It sketches the outline first.
And it assumes the middle is just more of the same until it hits another edge.
You've got it. And it does this using a hierarchy of different cell types. The text lists simple cells, complex cells, and hypercomplex cells. It's like building with Lego blocks.
Okay, let's walk through these because this is how we actually build a shape.
So you start with simple cells. These are found mostly in layer 4. And they're incredibly picky. They don't just respond to an edge, they respond to an edge at a specific orientation.
So one cell might only fire if it sees a vertical line. Another might only fire for a horizontal line.
Correct. And another for a 45-degree angle. If the line is tilted just slightly wrong, that cell stays completely silent. This is built by what we call linear organization. You have a row of excitatory cells that are flanked by inhibitory cells, so only a line that fits perfectly into that specific slot will trigger the signal.
Okay, so simple cells detect the angle of a line. They give us the raw geometry. What about complex cells?
Complex cells take it a step further. As the signal moves away from layer 5, the neurons get a bit smarter. A complex cell will respond to a vertical line, just like the simple cell. But, this is the key, the line can be displaced.
Meaning it doesn't have to be in the exact same spot on the retina.
Right. It can move laterally or vertically across its receptive field. As long as it's vertical, the complex cell fires. This is absolutely crucial for detecting movement.
Ah, I see.
If you're tracking a vertical tree trunk as you walk past it, the image of that trunk is shifting across your retina. Complex cells allow the brain to recognize it's still the same vertical line, even though it's hitting different photoreceptors from moment to moment.
And finally, we have the hypercomplex cells. These sound like they should be doing calculus or something.
They're definitely doing some serious geometry. These are in the outer layers. And they detect things like specific lengths or angles or shapes. A hypercomplex cell might only fire if it sees a corner or line of a specific short length. It detects the ends of lines.
So we're building the image from the ground up. First, we detect dots of light on the retina, then lines from the simple cells, then moving lines with the complex cells, then corners and shapes with the hypercomplex cells.
Exactly. It's a constructive process. We aren't just taking a photo, we are building a model of the world out of these geometric primitives. It's almost like how a computer renders a 3D video game, but in reverse. We're taking the pixels and figuring out the polygons.
And this processing splits into two major highways as it leaves the primary visual cortex. Which brings us to segment 5, the two major pathways of analysis.
We hinted at this with the Magno and Parvo distinction back in the LGN. Now we see the full realization of that split in the brain's actual geography.
Pathway 1, the dorsal stream, or as I like to call it, the action pathway.
That's a great name for it. This pathway goes from the visual cortex up into the parietal cortex, kind of the top and back of the head. It analyzes where things are and how they're moving, and it's built almost entirely on those magnocellular fibers.
So it's using that fast, black and white, low-detail data stream.
Yes. This is the pathway that lets you catch a ball, or duck a punch, or walk through a cluttered room without bumping into the furniture. It processes 3D position and the gross form of objects. It doesn't really care what the object is. It cares about its physics. It cares about its trajectory.
Okay, then you have pathway 2, the ventral stream, the identity pathway.
This one flows down into the temporal lobe, the side of the head. It uses the parvocellular fibers mixed with some magnofibers. And this pathway analyzes what things are. It processes color, texture, and fine detail. This is the part of your brain that is reading the words on this page right now or recognizing your mother's face.
It's amazing to think that recognizing a face and seeing where that face is in the room are handled by completely different parallel parts of the brain.
It leads to some fascinating neurological conditions. If you have damage to the ventral stream, you might develop something called visual agnosia.
That's a...
You can see a cup on the table, you can reach out and grab it perfectly because your dorsal stream is working fine, but you cannot tell me what it is. To you, it's just an object of undefined utility. You can't name it.
Wow, so that completely separates the seeing from the knowing.
Exactly. Conversely, if you damage the dorsal stream, you might have optic ataxia. You could look at the cup and tell me, oh, that is a cup. But if you try to reach for it, you'll miss by 6 inches every time. You've lost the spatial coordinates.
Wild. It really shows how modular the brain is.
Okay, let's move to segment 6. Binocular vision and depth perception. We touched on this with the stripes in layer 4.
Right. Stereopsis. The mechanism is actually simple geometry, but the neural implementation is incredibly complex. Your eyes are about 2 inches apart, right?
Right.
That means the image on your left retina is always slightly different from the image on your right retina. We call this disparity.
And the brain uses that disparity to calculate depth.
It does. There are specific neurons in the cortex that only fire when the images from the two eyes are not matching up perfectly, when they are, in a sense, out of register. The brain then calculates the degree of that interference and translates that into distance.
So it's literally measuring the error between the two eyes to figure out how far away something is.
That's a great way to put it. The text mentions this works really effectively up to about 200 feet or 60 meters.
And after that? After that, the disparity is just too small. The angle is too acute for the brain to measure accurately. Beyond 200 feet, we rely on other cues, perspective, relative size, shadows. But for close-up work, threading a needle, catching a ball stereopsis is king.
But this requires the eyes to be working together perfectly, and that brings us to strabismus.
Strabismus, or what most people call a cross eye or a squint. This is when the eyes don't fuse. It can be horizontal, vertical, or even torsional.
And the brain's reaction to this is pretty ruthless from what I read.
It is. If the eyes are pointing at two different things, the brain gets conflicting data. It can't fuse the images. So, to avoid the confusion and the double vision, it just suppresses the image from the repressed eye. It literally deletes the feed from that camera.
It just ignores one eye, it ghosts it.
Completely.
Yeah.
And if this happens in a child, that's where the real danger lies. There is a critical period for visual development.
Right.
If a child has strabismus and the brain suppresses one eye for too long, the cortical connections for that eye actually wither away. The neurons die off or they get repurposed for the other eye.
So the eye itself might be physically healthy, but the brain becomes effectively blind to its input.
Yes. It's called amblyopia, or lazy eye.
Yeah.
Even if you fix the muscles later in life with surgery, the vision might never come back to 20/20 because the software in the brain has been uninstalled.
So that's why early correction in kids is so vital. You're racing against the brain's own pruning process.
You are.
Speaking of muscles, let's talk about how we control these cameras. Segment 7, eye movements and control.
We have 3 pairs of muscles attached to each eye. The medial and lateral recti move it side to side. The superior and inferior recti move it up and down. And the obliques handle rotation.
And these are controlled by cranial nerves 3, 4, and 6. But the really interesting part is the types of movements we make. Because we don't just move our eyes, we have these very specific modes of operation.
The most fundamental is fixation, locking onto a target. And the source distinguishes between voluntary fixation and involuntary fixation.
So voluntary is finding the target, involuntary is keeping it there.
Exactly. Voluntary fixation is controlled by the premotor cortex in the frontal lobes. It's you deciding, I want to look at that bird over there. Once you find it, control hands off to the secondary visual areas in the occipital cortex for involuntary fixation. This mechanism locks the eyes onto the contrast of the object.
It's like a missile tracking system.
It is. Even if you try to look away sometimes, it's hard to break that lock. But here's the wildest part about fixation. Your eyes are never actually still. Even when you think you're staring perfectly at a single point, your eyes are constantly doing 3 things: tremor, drift, and flicking.
Okay, let's break those down. This blew my mind.
First, a continuous tremor. Your eyes vibrate at 30 to 80 cycles per second. That is incredibly fast. Second, a slow drift. The eyes just naturally drift off the target. And third, a flicking motion, or what we call microsaccades, that snaps the fovea back to the target whenever it drifts too far.
But why? Why can't they just stay still? It seems so inefficient to have them shaking all the time. Wouldn't that make the image blurry?
You'd think so, but it's actually essential. It's because of retinal adaptation. If the image on your retina was perfectly stationary, if the same photons hit the exact same rods and tones continuously, the photoreceptors would bleach out, they'd use up all their chemical pigment, and they would just stop firing. The image would literally fade to gray and disappear within seconds.
So you're saying the brain needs the image to jiggle?
Yes. The brain needs the image to jiggle constantly to keep the neurons excited. It needs the contrast to move slightly back and forth across the receptors to keep refreshing the signal.
That is incredible. We have to shake the camera to keep the film from getting bored.
Essentially, yes. If you could
okay.
perfectly paralyze the eye muscles so the eye was absolutely still, the world would just disappear. You would go blind.
Let's talk about the big movements now. Saccades.
Saccades are the jumps. Like when you're reading a book, you don't scan the line smoothly.
Oh, you jump.
You jump from word to word, jump, stop, jump, stop. When you view a scene, Your eyes jump from highlight to highlight a face, a bright color, a door. These jumps are called saccades. They happen 2 to 3 times every second.
But here's the mind-bending part. During the jump, you are blind.
This is the saccadic suppression we hinted at.
Yes.
The brain completely suppresses visual input while the eye is moving. Otherwise, every single time you looked across the room, you would see this dizzying, nauseating motion blur, like whipping a video camera around way too fast.
So the brain just cuts the feed, moves the camera, and then turns the feed back on when it's stable.
Exactly. So our visual reality is actually just a series of still snapshots stitched together so cleverly that it looks like a continuous movie.
That is amazing. And the text says 90% of your time reading is spent in fixation. Only 10% is the jump.
And you don't perceive any of the gaps. The brain just edits them out. It stitches the timeline together seamlessly. You are technically blind for a significant portion of your waking life, and you never, ever notice.
Then we have pursuit movement.
This is different from saccades. This is when you track a moving object, like a car driving by. The eyes lock on and move smoothly with it. This requires a highly developed cortical mechanism to calculate the trajectory and speed of the object and match the eye muscle speed to it. It's actually much harder for the brain than a simple saccade.
And finally, the superior colliculi make another appearance. The source mentions their role in turning.
Right, the superior colliculi are responsible for that reflex turn toward a visual disturbance. And here's a fascinating clinical note. This works even if the primary visual cortex is destroyed.
So a person could be cortically blind, unable to see consciously, but if you flash a light to their side, their eyes will still turn toward it.
Yes.
Yeah.
And they won't know why they looked. They won't see what is there. But that ancient part of the brain, the lizard brain, still works. It still directs the eyes. The phenomenon is called blind sight. It's one of the most eerie and profound things in all of neurology.
That is spooky. It really drives home that old system versus new system idea. Okay, final segment. Segment 8, autonomic control, focusing, and pupils.
This is the automatic adjustment of the camera settings. Let's start with the combination, which is just focusing. This is controlled by the parasympathetic nerves.
And the mechanism.
The ciliary muscle, little ring of muscle inside the eye, contracts. This relaxes the ligaments that are holding the lens. And a lens, which is naturally elastic, snaps into a more spherical, thicker shape. This increases its refractive power for near vision.
So to look at something close, your eye muscles actually have to work harder.
Yes. That's counterintuitive, isn't it? Relaxing your eye lets you see far away. Flexing your eye muscle lets you see close up. That's why reading for hours and hours causes eye strain. Your ciliary muscles are basically doing a bicep curl for 3 hours straight.
But how does the eye know how to focus? How does it know if an object is blurry because it's too close or too far? I know a camera uses contrast detection. What does the eye use?
The text lists some incredibly clever clues. One is chromatic aberration. Red light and blue light bend slightly differently as they pass through the lens. The eye can tell which color is in better focus and use that to calculate distance.
No way.
Yeah. Another is oscillation. The lens actually vibrates slightly, testing if making it tighter or looser makes the image sharper. It happens so fast you don't notice it.
Then we have the pupils, the aperture of the camera.
The pupillary light reflex. Light hits the retina, a signal goes to the protectal nuclei, then to the Ehringer-Westphal nucleus, and then back along a parasympathetic nerve to the iris sphincter to constrict the pupil.
And that's called meiosis.
Meiosis. It's a protection mechanism, but it's also for adaptation. The pupil can change the amount of light entering the eye by about 30-fold. You combine that with retinal adaptation, and our eyes can handle a huge range of light intensity, something like 500,000 to 1, from the darkest night to the brightest sun.
Finally, the source mentions a condition called Horner syndrome. This is what happens when this autonomic control breaks.
Specifically when the sympathetic nerves to the eye are interrupted. The sympathetic nervous system usually dilates the pupil and helps keep the eyelid open. If you cut that nerve chain in the neck, you get Horner syndrome.
And what does that look like?
It's a classic diagnostic triad. You get constricted pupil because the dilator muscle is paralyzed, a droopy eyelid, which is called spitosis, and interestingly, no sweating and flushed red skin on that one side of the face.
Wow.
If a doctor sees that combination, they know exactly which nerve pathway is damaged.
It just goes to show how integrated vision is with the rest of the body's systems. It's not an isolated thing at all.
Not in the slightest. It connects to your spine, your midbrain, the cortex, the autonomic system, the muscles. It's everywhere.
Okay, let's wrap this up. We have traveled a long, long road today. I mean, we have traced a signal from a photon hitting the retina through the chiasm where it gets started, into the LGN where it gets filtered and gated,
Hmm.
up to the cortex where it gets sliced and diced by simple and complex cells, split into where and what pathways, and then finally reconstructed into a 3D reality.
It is a massive amount of processing. It accounts for a huge percentage of our brain's total energy consumption.
What is the one thing you really want the listener to take away from this entire deep dive?
For me, I think it's the concept of the construct. We tend to trust our eyes implicitly. I saw it with my own two eyes.
Yeah.
Right. But as we've learned today, the brain ignores steady signals. It suppresses vision entirely during movement. It fills in our blind spots. It creates depth from a mathematical calculation. The visual world you experience is a construction. It's a very good construction, one that's vital for survival, but it is not a raw feed of reality. It's an edited story.
That is a provocative thought to end on. We are all just walking around watching our own personal brain-edited movies of the world.
And luckily for us, the editors are award-winning professionals.
Indeed. Thanks for listening to this deep dive into the eye and the brain. We'll see you, or rather, our brains will construct a representation of you next time.
See you then.